Hi, my name is Nissa Thompson, and in this presentation I'll be discussing how Tozuleristide is used in identifying gliomas. What are gliomas? Gliomas are a tumor on the brain and spinal cord that originate in glial cells. These are the cells that support the main nerves. There are three types of gliomas called astrocytomas, ependydiomas, and oligodendrogliomas. Gliomas are extremely serious in that they affect brain functioning and can be life-threatening, and often are fatal. The nature and treatments for the types vary depending on which of the three it is, but they generally include surgery, radiation therapy, and chemotherapy. Gliomas consist of approximately 33% of all brain tumors. Something that makes them particularly difficult to treat is how they become deeply infiltrated into healthy brain tissue. Especially along the edges, they have tentacle-like projections that become embedded deep into the healthy brain tissue. They become hard to distinguish, and they are not very responsive to chemotherapy or radiation treatments. Therefore, surgical removal is often the best option for patient survival. So, when you're identifying gliomas, visualizing them is most important to surgery. Therefore, fluorescent-guided surgery has become popular. With FGS, fluorescent-guided surgery, it pairs fluorescent contrast agents with detection devices. Something that is being used to help identify gliomas is Tozuleristide, also called BLZ-100. It's a synthetic non-toxic form of scorpion venom. It's a near-infrared imaging agent that is composed of chlorotoxin and has near-infrared fluorophore indocyanine green. The chlorotoxin bonds well to various types of human tumors in other experiments prior to this one. So in this study, 17 subjects were involved and they were administered intravenous doses ranging from 3 to 30 milligrams. The fluorescence was observed in situ, which means during surgery, and in ex vivo, for which the tissue sample was removed for later observation. They concluded that no dose-limiting toxicity was observed, no adverse effects were reported, and the Tozuleristide was well tolerated in doses up to 30 milligrams. The imaging shows that contrast could be observed within hours of dosing, and there's a strong potential to highlight residual cancer, and further clinical trials are justified. The fluorescent dye can be seen in these 6 pictures, in which the green is the highlighted tumor. These are my references. Thank you.